morning, viewer. Welcome to our Glory Globe TV. Here's a breaking and most amazing news for our viewer. Recent neuroscience studies have consistently outlined the role of C90RF72 gene in the development of some neurodegenerative disease. These studies found that mutations of this gene increase the risk of developing amyotrophic lateral sclerosis, that is called LAS, for frontotemporal dementia (FTD), two neurodegenerative disorders characterized by motor impairments, issues with communicating, and other distinct symptoms. Researchers at Johns Hopkins University School of Medicine, University of Chicago, the Howard Hughes Medical Institute, and other institutes in the United States recently carried out a study aimed at better understanding the processes through which C90RF72 gene mutation might ultimately contribute to the development of ALS and FTD. Their findings, published in Nature Neuroscience, highlighted a promising avenue that could improve therapeutic interventions for this complex neurodegenerative disorder. My research group has been interested in identifying modifiers of the C90RF72 repeat RNA metabolism. Dr. Sayang Son, one of the researchers who carried out the study, told Medical Express a hexanucleotide repeat expansion in the C90RF72 gene is the most prevalent genetic cause of both ALS and FTD. As the expansion is located in the non-coding region, the repeat containing RNA is believed to be one major factor that drives the disease pathogenesis. Sun and her colleagues have been trying to identify genetic modifiers of the unconventional production of toxic polydipeptide proteins from the repeat RNA. This could in turn allow them to identify viable strategies to prevent the accumulation of these toxic proteins and thus reduce their toxic effect. In one of their previous studies, they thus performed a genome-wide CRISPR-Cas9 screen to search for genetic modifiers of dipeptide production. We found that the two M6A methyl transferases or candidate genes that can modulate the polydipeptide level. Sun explained, we also analyzed public proteomic data and found the two methyl transferases are downregulated in C90RF72 ALS/FTD patient iPS neurons. Altogether, this initiated our further study on the M6A dysregulation and how this contributes to neurodegeneration in C90RF72 ALS/FTD. Sun and her colleagues performed a series of analysis on patient-derived induced pluripotent stem cell, that is called PSC, differentiated neurons, and post-mortem tissues extracted from deceased patient with ALS/FTD with C90RF72 gene mutation. Using various high thought-out genetic sequencing techniques, they specifically assessed as an RNA modification known as M6A and its impact on the regulation of genes across the full range of expressed messenger RNA known as transcriptome. We also examined the molecular mechanism of M6A regulation on C9 repeat RNA. Sunset, finally, we measured neuron survival to examine the rescue efficacy by modulating the M6A pathway. Our findings reveal a novel layer of RNA regulation that plays a critical role in the pathogenic mechanism underlying neurodegeneration. Overall, the findings gathered by this research team showed that the M6A modification in the C90RF72 genetic sequence of expanded repeats facilitate the decay of RNA via the nuclear reader YTHDC1, thus regulating the repeat. Moreover, reduction in M6A appeared to facilitate the accumulation of repeat RNAs and the encoding of toxic proteins known to be associated with neurodegeneration. Be in the report.